Hello, everyone, and welcome to the Molecular Devices webinar series. Today, I'm pleased to present to you Dr. Sam Deutsch, who will be speaking to us on the synthetic metagenomics, converting digital information back to biology. And now, Dr. Sam.
Good morning, everybody, from Walnut Creek in California. Thanks to everyone who's attending this web-based seminar. So as Natalia said, today I just want to give you an overview of synthetic biology at the TGA, which for us is mostly about converting digital information that has been generated through sequencing back into biology. So I will break my talk into two sections. First, I want to give you a vision of how we see synthetic biology, a few slides on the operation that we have set up with some metrics. And then the second part of the talk, I want to go over some examples of science projects that just give you a glimpse of how we are using synthetic biology to enable new types of science. So we are at the TGA in California, and we are the biggest non-medical sequencing center in the world. We do very diverse type of sequencing projects, ranging from the more classical bacterial isolates or plants and fungi. So we are the biggest sequencing institute for plants and fungi. And then all the way to more novel types of sequencing approaches where instead of looking at one individual cell or organism at a time, we try to sample whole environments. We call that metagenomics or metagenomics. And also we have a very extensive single cell program where single cells are individually sorted and sequenced. And this is very useful in cases where it is not possible to culture cells. And we know that about 99% of the cells we don't really know how to culture them. So doing single cell genomics really opens a complete new space in terms of sequencing. So over the past 10 years or so, we've been generating this really huge amount of sequencing. We generate about 100 bases of sequence every year. And sequence that we generate as well as the sequence that is generated elsewhere is just feeding this very massive sequence databases. There's a lot of activity also in database development. So we're developing tools that allow us to very efficiently query databases, perform different levels of analysis, and try and start generating hypotheses about the different biological activities that happen within an organism or within an environment. But moving from these sequence databases into real-life applications, for example, for trying to develop things that we can use to solve real-world problems is really limiting. And part of the problem, at least how we see it, is that we have two very different types of information. On the one side, we have digital information in databases that we can very easily access. And on the other side, we need biological molecules that we can assay for function. And going from one to the other is not always true. So the reasons for this is, in many cases, we don't have easy access to templates. This is particularly the case when we're looking at metagenomes or single cells. So we destroy the single cells in the process of sequencing them, so it's not very easy to find an identical cell that we can use as a template. But in many cases, even if we did have a template, the native sequence of some very exotic organisms might not actually work in the model organisms such as E. coli and yeast that we're used to working with in the lab. Maybe the codon usage is very different and will lead to very poor expression in our model systems. And when we're dealing with not a gene, but multiple genes in a pathway or an operon, transferring this sequences as they are usually doesn't work either in the model organisms. And that's because regulatory sequences and regulatory elements don't transfer very well, and also because there's a lot of cryptic regulation that we really know very little about. So synthetic biology actually offers a bridge between this two types of information. So synthetic biology, at least in this sense, is about rewriting the DNA in a way that maximizes the chances that it will work in your model organism. So at the TGA, our vision is to have an integrated pipeline that goes all the way from biological questions that are generated by users. A lot of these questions can be answered through sequencing data, not only genomic sequencing, but also perhaps transcriptome sequencing or transposon bombing, or ChIP-seq, or lots of different information that we can generate with the sequencing instrument. Once the sequence data is generated, we need to perform different levels of computational analysis. For example, the sequences from the short read instruments need to be assembled into longer configs. Then we can perform annotation steps, and we can do a lot of functional predictions to kind of try and determine what some of the protein-coding regions in that sequence might be doing. The next step would be, now that we've generated all this hypothesis, we would want DNA synthesis to allow us to validate experimentally this hypothesis in a large-scale manner. Synthesis, as you might know, is a very error-prone process, so we need not only to synthesize, but also to do QA and QC on the synthesis that we generate, just to make sure that the things that we're producing are what we want them to be. And then we can put them into vectors, and we do a lot of vector design with our users. So once that the synthetic parts are in the vectors, they can just be plugged in into our user's pipeline for high-throughput functional verification. Sometimes we do functional screens at TGA, but in many cases we synthesize for our users, and our users have their functional validation screens in-house. So over the last couple of years, our synthesis program is about 2 years old. We've been putting a lot of work into developing a very streamlined pipeline for synthesis. So as I said before, everything starts from databases. And then we've put a lot of effort into developing different design algorithms. So for example, we have a piece of software called Gene Design that allows you to take any gene, decide how you're going to partition it, decide how you might want to code optimize it for different hosts, and then come up with a strategy for synthesis. J5 is the next step along the design pipeline. So that is, once you have different synthetic parts, how are you going to put them together to build bigger devices? And we also have a very unique tool called BioStudio that allows you to take a very high-level view. So for example, if you want to design an entire chromosome or even an entire organism, how would you go about that? So basically, we have a pretty integrated design pipeline that allows us to go from the sequence databases to our synthetic design. So then we just push a button, which will in effect order a set of oligos that we will use for the molecular biology parts of the synthesis program. In terms of the oligos, we work with essentially two types of oligos. Oligos that are synthesized directly on an array that then need to be cleaved and be conjugated. But we also work with the more traditional types of oligos that are synthesized in columns, and they come in 3 different web types. So once we get the oligos either from an array or in plates, then everything downstream is the same. So we have a set of liquid handling systems, both the traditional robotics type that work in pretty large volumes, 20 to 25 microliters. We're starting to move everything to 1 to 2 microliter scale volume using this new generation of liquid handling systems called acoustic printers that allow us to manipulate even drops of 2.5 nanoliters in size. Then we perform all the molecular biology, which involves assembling the oligos into larger pieces and cloning them into our vectors and transforming them into E. coli, which is our intermediate host. And then once we do transformation, we use colony depleters and pickers for the QPIC series from molecular devices, which allows us to in effect handle thousands upon thousands of clones every week. So the clones get transformation get plated. We pick about 8 clones per every synthetic fragment that we want to produce. And everything from a batch gets pooled, and we sequence using the next generation sequencing technologies, which has the advantage of a very fast turnaround. So we usually get our data back in 3 days, and the analysis is very robust. So there's very little manual intervention here. We're just basically out of our sequencer. We have a suite of analysis tools that just basically generate a table, and for each part we can identify which clone, if any, is exactly what we need. Then basically we have to move our fragments into our final destination vector, and then they can be used for the high throughput functional characterization studies. Next slide gives you an idea of the kind of throughput that we're dealing with at TGI. So the red graph shows you our output per quarter in terms of 1 kb sequence verified parts, which is our essential unit for synthesis. And you can see that in our last quarter, we generated just over 700 kb synthetic sequence, and for the next quarter, our target is to generate about a megabase. And our cycle time, which is shown in blue, is in weeks, shows you that we've been able to decrease our cycle time, so that it takes us about 2 weeks to generate a batch of between 100 and 200 1 kb synthetic parts. Next slide shows you an evolution of what our users are asking us to deliver. So when we started at the beginning of last year, most of what we were producing were in gene size fragments. So most of our users requested synthetic parts that were 1 to 1.5 kb in size and usually comprised a single gene, which our users or ourselves wanted to test for a particular function. As time has gone by, we see that our users are kind of changing what they want, and more and more we're getting requests for larger constructs. As you see in Q4 of last year, a lot of our requests were for fragments that were 5 kb or larger, which usually encompass multiple genes that work together for a biological function. Our biggest projects to date are around 50 kb. And that's kind of the niche where we want to be at TGI. We want to be rather than making single gene fragments that can also be provided by industry. We want to concentrate on larger genes, on the generation of combinatorial libraries, on large, even multi-operon devices that will really have an impact on the phenotype of the final expression host. Other very important consideration is cost. As you all might know, the cost of sequencing has gone down very dramatically over the last few years. But the cost of synthesis, even though it has gone down, it's been more linear than exponential. And when we started our operation in 2011, it cost us about 50 cents per base pair of synthetic DNA that we produced. That was pretty much in line with the industry costs. We've been able to decrease, so our current cost is about 18 cents per base pair, and we see this going down. And we're hoping that by the end of this year, beginning of next, we're going to be significantly below 10 cents per base pair. But this is still pretty expensive. If you think that some projects require synthesis of millions of base pairs, it's still a substantial cost. And we think that the main drivers of our cost reduction are going to be new, cheaper sources of oligo, and also moving all of our reactions from the current microliter scale to the nanoliter scale using solutions such as the printer that I mentioned. So for this year, our target is 3.3 million bases of synthetic DNA, and it has all been allocated. So about 2/3 of that goes to our super users, which are these very large bioenergy centers that are really focused on energy and environmental issues. But we also have every year a call for projects. That's the CSD area here. So this year we did 800,000 synthetic DNA for this project, and basically anybody can apply, and these projects are reviewed externally for scientific merit as well as DOE relevance. So we're looking for things that address energy or environmental problems. And this part of the top part of the pie chart shows you our internal projects. So what are we using all this capacity for? So this slide shows you just I just want to give you a flavor of the type of projects that we work on. For example, we still do a lot of single gene characterization, but we're also moving more and more into pathways, which so we can generate synthetic pathways, and we can generate synthetic pathways also combinatorially. So we we start from a few sets of genes, but because we can stitch them combinatorially, we end up with a very large space of synthetic pathways that we can actually function. We are also putting a lot of attention into creating safe and versatile chassis organisms where we can use our synthetic biology studies. So safety is a primary concern. We want to do things in a way that will minimize the risk to the environment. And there's a lot of negative public perception around the field of synthetic biology, and we want to be very proactive in communicating to the community that safety really is a primary concern. And then at the bottom, show you an example of going all the way. So at the top, I'm showing you gene characterization, pathway characterization, all the way to this synthetic Saccharomyces cerevisiae 2.0 project, which is basically resynthesizing the whole genome of yeast. So I will give you more details on two of these projects and also give you an idea of what we're doing in terms of safety. So the first project was a collaboration with the Joint Bioenergy Institute at Emeryville. And one of their main goals is to develop the field of cellulosic biofuels. So one of the main limitations for the development of cellulosic biofuel is the lack of sufficiently active and industrially robust enzymes for the breakdown of biomass. And at the time, Blake Simmons, a collaborator for this project, was particularly interested in one type of enzymes called GH1, which participates in the final step of biomass breakdown, whereby cellulose is converted into glucose. And they had some of these enzymes at hand, but they needed an enzyme that would really fit the industrial process that they were trying to develop. So they wanted an enzyme that would have good activity at 70 degrees Celsius, that would be stable at a pH of 4.5, and most importantly, that an enzyme that would work in the presence of ionic liquids. So ionic liquids is a pre-treatment method that they use. So as you might know, biomass is usually very recalcitrant to enzymatic activities. So you have to do something to make the sugar more available to enzymatic digestion. So what they're using is ionic liquids. The downside with ionic liquids is that it's pretty toxic to enzyme activity. So they wanted to see whether we could come out using synthetic biology with an enzyme that would fulfill all these three criteria. So the approach we took was, I call it synthetic metagenomics, which was basically to go out there, see what nature has done in terms of diversity. So we just pulled out from the databases anything that looked like a GH1 enzyme, and then we just tried to sample systematically this phylogenetic diversity. So what I'm showing you here is a phylogenetic tree of about 1,500 GH1 enzymes that we found in databases, and we also had some internal data that we had recently generated. So just a month before we started this project, we had completed this rumen study in which the metagenome of the cow rumen was sequenced and outlined, and from there we were also able to identify about 50 additional GH1 enzymes that were not available in public databases. So in this tree, you see that we identified enzymes from all kingdoms of life. So everything that is in red is an enzyme that comes from bacteria. Everything that's in blue comes from eukaryotes, that means plants or fungi or protists. The things in purple are the things that we identified from the cow rumen, and the things in green are from these archaeal organisms, which are usually a very extreme type of prokaryotes. One interesting thing to note is that from the rumen enzymes, the things in purple, even though there weren't so many, they contributed very significantly to expanding the phylogenetic diversity. So for example, you see up here, this very interesting part of the tree, you see that some entire branches of the tree are defined by this enzymes that we identified in the rumen. So and this is kind of a recurrent thing that we find, that if you want to increase your protein diversity, metagenomics is a very good way to do it. So we have this big tree, and we wanted to sample it in an unbiased way. And these are these red dots that I show in the outer circle. So these are the enzymes from the tree that we sampled. And you can see that some areas of the tree, for example, here, almost every representative was selected. And that's because you see that this area of the tree has very long branch length, meaning that every enzyme is very different to each other. Whereas other parts of the tree, for example, here, has pretty short branch length, meaning that one representative is pretty much enough to capture all of the diversity in that part of the tree. We try to do this, as I said, in a very systematic and unbiased way. So in total, from the 1,500 genes, we selected about 200 for synthesis, including 15 from the cow woman. So we went ahead and did the synthesis, and for about 180 out of the 200, we were able to successfully complete both the synthesis and the cloning into the expression vector. And then we put this into a host and induced overexpression. In about 65% of the cases, we were able to identify 50 kilodalton protein, which corresponds to the enzymes that we were looking for. Next, now we have this proteins in tubes. We could go ahead and start doing some functional characterization. This gives you the first results that we got from the first 15 enzymes that we were able to purify. And what I'm showing here is the enzyme activity at different temperatures. So, for example, if you look at the case of enzyme 159, you see that its optimal working temperature is 50 degrees Celsius, but once you go above 50 degrees, almost all that activity is lost. But we also see other enzymes, such as enzyme 22, that was very active all the way from 30 degrees Celsius to 90 degrees Celsius. So it was a very, very thermostable enzyme. So this is the kind of enzymes that we were looking for in terms of them being very good candidates for a biotechnological drug setting. So our study design for this project called for all of our 180 GH1s to be characterized as activity at least 4 different temperatures, 2 different pHs, against 3 different substrates, and everything done in 2 replicas. So it required about 15,000 reactions to be performed. And doing this in the traditional low throughput volumetric plates was taking us quite a long time. We were lucky. So we needed a very high throughput screening method in order to do this in a timely fashion. We were lucky that Trent Northern at Lawrence Berkeley Lab was at the same time developing a method to do just this. So his method is called nanostructure initiator mass spectrometry. So in essence, what we can do is spot the enzyme substrate reactions at different conditions into a solid support. And each of these very small droplets that get printed on the array can be interrogated with a laser. So the laser will hit each one of the spots, causing the reaction to ionize and generate a mass spectra that is very quantitative and allows us to calculate the rate of conversion between our substrate to the product. So this next slide I'm showing you some actual NIMS data that we got from this project. So each of the dots, as I said before, corresponds to a single reaction, and the color corresponds to the amount of conversion that we observed. So the lighter the color, the more conversion. So everything that you see in white means that almost 100% of the substrate was converted to product. So this is what the actual spectra that corresponds to each of these dots looks like. So for example, in this case, you see you have cell-biased substrate, and a significant proportion of that was converted to glucose, which is the cell-biased product. We were interrogating multiple substrates at the same time, in multiplex. For example, in this case, you see that we have a maltose substrate, and we don't see a maltose product. So that particular enzyme was very active against cell-biased, but was not at all active against maltose. So we could summarize all of our 15,000 reactions in a single figure. So this is can be plotted back to the tree. And this tree now shows you only the 200 representatives that we decided to synthesize. As you see, it reproduces very well with the topology of the original tree, even though now we have only 200 representatives instead of the initial set of 1,500 enzymes. And then we have 4 tracks at the outer circle. So the first one shows you the expression, so everything in black means that the protein was expressed. And then we have activity against the 3 different substrates, cell-biased, lactobacillus, and xylobacillus. And the color represents the maximum temperature at which we observe activity. So for example, this enzyme over here was active against cell-biased and lactobacillus all the way up to 90 degrees Celsius. Whereas you see other enzymes, this part of the tree, for example, that are only active at 40 degrees, and if you increase the temperature any further, you lose all of activity. So this tree has a lot of information, a lot of very interesting information. For example, you see that there are still enzymes, as we expect, are very enriched for thermostable enzymes. But we also find pockets of thermostable enzymes elsewhere in the tree, sometimes where you expect them to be, other times you wouldn't necessarily expect them to be. So it just validates that taking this unbiased metagenomic approach really gives you a lot of information. Also, we found that the whole area of the tree around here, we saw very, very little activity, and we've just very recently figured out why this is the case, because these are enzymes that recognize only phosphorylated versions of the substrate. That's also a very interesting finding, and they tend also to cluster in one side of the tree, and we've actually identified a single nucleotide that is responsible for this preference for one type of substrate versus another. As I said at the beginning, we're looking for enzymes that were thermostable, but also for enzymes that would resist ionic liquids. So next, we took a set of about 15 enzymes that were shown to be highly thermostable, and then we tested them for activity in the presence of ionic liquids. And we did this both with a 10-minute pre-incubation and with a 24-hour pre-incubation. And the reason we do 24-hour pre-incubation is that if you want an enzyme to work in an industrial setting, you want an enzyme that will really work over the course of fermentation, and this fermentation typically take 1 to 2 days at least. So with a 24-hour measurement, what we're actually looking is that how much activity is left after 24 hours. And those are enzymes with high residual activity is what we're really looking for. So in the blue bars is just the initial baseline temperature after just 10 minutes of incubation, and the red bars after 24 hours. And you see that in at least 5 cases we still have substantial activity after 24 hours. So basically we identified 5 enzymes that completely fit the specifications that we were given when we started this project. Interestingly, 3 of the enzymes clustered to one part of the tree, but we also see some very good enzymes that are elsewhere that also perform very well. Again, validating this unbiased phylogenetic approach. Just to summarize our conclusions from this study, we synthesized 180 of these diverse enzymes. They were all characterized in terms of cellular expression, activity against different substrates at different pHs, temperatures, and activity under ionic liquids. No previous enzymes were known to work under these required conditions, and then after performing our synthetic metagenomics PET study, we're able to identify 5 that will fulfill our criteria very well. And now our partners at JV are further studying the potential use of these enzymes in an industrial setting. Now I want to switch gears to a different type of project. So whereas the first type of project we're looking at the activity of single genes, now we want to address how different genes work together for biological outcome. And in this case, we were looking at polypeptide synthases, which are very common type of pathways that are used for the production of secondary metabolites. So a lot of, I think at least 50% of the drugs in the pharma sector are derived from polypeptide synthase type pathways. So everything, most of the antibiotics and a lot of the anti-cancer cells and immunosuppressants are PKS or NRPS derived molecules. And in essence, these PKS pathways are a biological assembly line. So you see all of them will have a loading domain, a variable number of extension domains, and a termination domain. And the loading domain will take a molecule that's common to central metabolism, so something typically like malonyl-CoA or acetyl-CoA, and will load it and covalently bind it, and then it will pass it to the first module downstream, and each module will perform a set of chemical transformations. So usually it will involve extending the carbon chain and doing oxidations or reductions to basically modify the chain as needed. And there's a final termination module which has thioesterase activity that will usually cleave the molecule and perform a final level of modification. In this case, it's cyclizing the molecule, so it becomes a closed molecule and they tend to be usually very potent active biological molecules. So if we look at databases and also especially our own internal metagenomics databases, we can identify a very large number of these modules which are part of TGA pathways. And we can also predict bioinformatically what kind of transformations they carry out. And because TGAs are modular, you feel that you could just stitch them together in different combinations to generate a very diverse set of final molecules. In practice, what happens is that these modules, they really have to talk to each other very well. You just can't stitch them and expect the molecule to be passed covalently from one model to the rest. So only certain combinations of these modules will work very effectively, and that's what we need to find. So in our case, we're starting with a pretty simple test case where we just want to stitch 3 of these modules together. And our desired output for this are short chain, typically C4 to C6 hydrocarbons that can be used potentially as biofuel replacements. So for this pilot project, we synthesized 10 versions of the loading modules, 10 versions of the extension modules, and 10 versions of the final termination module, and we can stitch them combinatorially. So you can just synthesize it individually, but let the assembly proceed completely randomly using Gibson-type assembly technologies to generate ideally about 1,000 different pathways, and each of them individually would need to be, first of all, QC, so that to make sure that the sequence is what we want the sequence to be, and that poses certain challenges. And also it needs to be characterized in terms of function. So what, if any, target molecule is being produced. And the classical way to do this is using GCMS methods, which are very low throughput. So one of the challenges for this project is to find a way to go from the low throughput paradigms to high throughput paradigms. And one way that we think we can do this is through the development of biosensors. So instead of you having to measure the production of a target molecule, you get the cells to tell you themselves how much of this molecules have been produced. And this is an example of a biosensor that we recently synthesized. It's about a 15 kilobase device that has 3 different components. So it has a component a processing unit that actually involves 5 different genes. So it takes what the cell is producing and converts it into an alcohol. And we need to have an alcohol because we have an effector molecule called, in this case, DMOR, that will bind specifically to short chain alcohols. And when it binds to the alcohols, it will then bind to a promoter and activate a downstream GFP, which is telling us that the target molecule has been produced. And the level of GFP is also correlated to the amount of the target molecule that the cell is actually sensing. So once we have GFP produced, if we're lucky, then we can use downstream detection platforms to quantify large numbers of cells to tell us which of these cells are actually producing our target molecules. So this is an example of how this synthetic biosensor that we produce is functioning. So in this case, you see the amount of fluorescence when no ligand is available. And if we add ligand, in this case a C6 hydrocarbon, to the cell culture medium, you see that a lot of fluorescence is being generated, and even at pretty low levels of our target molecule. So in terms of the detection platforms, we are working with 2 different detection platforms. One is a FACS-based approach, where we first encapsulate the cells, let them grow, and then induce them for our pathways, and then we can sort them, and if any of our cells becomes fluorescent, it will be a very good candidate for the production of the ligand that can be then further characterized using more standard technologies such as GCMS. Alternatively, we're also working with our fluorescence colony imaging picking instruments, whereby the cells can be plated in a medium that uses the production of our pathways, and then fluorescent cells can be imaged and taken and transferred using the QPix 400 series capabilities. And the reason why both of these technologies are advantageous relative to standard FACS is because it allows you to clonally isolate the production of each individual cell. So a lot of these target things that we're trying to produce will diffuse. So if you just do the screens in liquid, the target molecules will be diffused and uptaken by a neighboring cell, which might also generate an activation signal, but it's not related to its own production, but the production of its neighbor. So we really need to find a way to isolate the reactions from one cell to the next. So both a droplet-based FACS as well as the colonies on the plate will achieve that. And the second advantage is that instead of looking at the activity of a single cell, you're looking at the activity of a colony or a droplet where you have multiple cells, and you get much more robust signals that are really the average of the population, not just a single cell. So as a summary for this part of the talk, we have completed the synthesis and assembly of our modules and verified that we actually generated a very complex library. Almost 100% of our expected targets were generated. We also synthesized a device that allows to sense endogenously the production of our target. Because the synthetic devices are in the order of 10 to 15 kb, the QA/QC is challenging, but we've developed solutions using our next generation sequencing platforms and software and analysis pipelines that we have written to assay both the connectivity in terms of the complexity of the libraries that we generate, as well as the sequence identity to make sure that the pathways synthetic pathways that we generate don't have a lot of errors. And then I talked to you about the different platforms downstream that we can use for detection of, and that we can do this in a very high throughput way. The last, I only just want to give you a few slides of how we're thinking about safety and the development of safe chassis. For us, the definition of a chassis is a material system that has been engineered with features that enable it to serve as a safe and convenient platform for subsequent synthetic biology manipulation. And the features that we would like this chassis to have is that we would want its DNA not to be transferred to other organisms, and even if they are transferred, we don't want other organisms to be able to read what those synthetic genes are. We want our chassis to be robust, so it can be used in an industrial setting. And one of the main problems we encounter in industrial fermentation are phage infections. It would be great if we could have a cell that's resistant to phage infections. And we would want the growth of our cells to be restricted to the laboratory. So if, by chance, the cells were to escape, they would not be able to survive. So we have two levels of safety. So one is organism level safety, so if the organism itself can survive, but also safety at the DNA level. So even if the DNA gets out, it won't be properly understood by other organisms. So to build this chassis, we're starting with an organism called Codon Depleted C. And we chose the organism because it has a relatively small genome, it replicates fast. We understand quite a lot about its metabolism. It's transformable, so we can turn it into a genetic model organism. And also, it's very robust industrially, so it's currently used by industry for the production of amino acids. So there's a whole industry built around this organism. So the first step for us to be able to convert the glut into a safe chassis is that we want to kind of change its genetic code. So the first step to doing that is that we're going to remove one of the termination codons, so this TGA Opal codon, and there's about 400 instances of that codon in the genome of Green Botanicum. And we are going to also take out the machinery that interacts with this codon, and then we're going to add an orthogonal translational machinery so that this codon is interpreted as something else. In this case, we're going to change it, for example, to an arginine. So instead of reading a subcodon, we will read an arginine, and then we can put this new subcodon in all of our synthetic genes. So if the synthetic genes get out, other organisms will read that codon as a stop and will not generate viable proteins. And then we can also put this new codon in essential genes within Codon Depleted C. So if the orthogonal translation machinery is not expressed, and we can put that machinery under an inducible system that you will only find induced in the lab. So unless you can properly translate essential genes, you will not be able to survive. So if the bug ever gets out, you will not find the inducer, you should not be able to survive. So I'm not really going to give you a lot of details on how we're doing this right now. Just to say that we're using this new technology called Cas9 CRISPR that allows us to do very targeted modifications of the genome and to do this at scale. This slide here is, as we get to the end of this presentation, I just want to kind of summarize, at least in our view, what we see are the important steps for developing DNA synthesis and synthetic biology so that you can really have a high impact in enabling a new type of science. So we need to have efficient synthesis pipelines. We need to be able to generate both short fragments like gene size, but also much longer fragments. For example, we want to design and synthesize a chromosome or large operas. We should be able to do that. We need to be able to do this combinatorially. And we also want to be able to do targeted modifications of the genome. And these are all areas that we are addressing in our environment. We need to be able to perform very compelling science. And we don't think that all the science should come from us, but should also come from the interaction with our users. I think we're very lucky to be in the Bay Area, which is a very active ecosystem in the field of synthetic biology. We have very good and exciting interactions with different players in the field in the Bay Area, but also we have close interactions with our BRCs, so the bioenergy centers that are located in different areas of the country. And we're also very interested in developing more and more interactions with anyone that comes up with a very interesting idea. So that's why we have this community synthesis projects that we also support. And the next thing that's very important is that we need to be able to do this in a very safe and reliable manner. If there's the public perception that this is very dangerous and we shouldn't be doing it, the consequence is that it's going to be shut down. So, PR and effectively communicating both the safety as well as the benefits of this type of science is very important for us. And finally, although I didn't speak a lot about this, I think it's essential that we develop very good tools for computer-assisted design and synthetic biology. This is going to become more like engineering rather than just biology. And having the tools that allow me to do this is going to be critical. So my last slide before I end is, as I said, we're very interested in having projects from the community, and for this we have this CSP or community sequencing or synthesis projects. And in terms of synthesis, what we're looking for is projects that address refactoring, screening, and functional characterization of multi-gene pathways involved in DOE mission relevant areas. We will still consider all the type of projects, but those are the kind of projects that we are encouraging. So things that involve the synthesis of large DNA molecules and also ideally would exploit some sort of coupling with very high throughput chain technologies would be very exciting to us. If you want more details on this call, you can find them at this URL here. For next year, we are looking to fund support perhaps 5 to 8 different projects, each one with a synthesis allocation of about 250 capable projects. And the deadline for this letters of intent are very soon, so next week, on April 8th. So with that, I want to thank you all for your attention, and we'll be very happy to take questions.
Thank you very much, Sam. So I think at this point we are ready to take any questions. So please enter your questions in the Q&A box, and Sam will be happy to answer those as we move further. Thank you.
So I think there is a question coming in from Colton Hicks. So do you think the majority of cheaper oligonucleotide synthesis platform will come out of research institutes or commercial group? And why? So I think, The development of nucleotide synthesis technologies are mostly industry-driven right now, and it's not necessarily a field where a lot of public institutions want to be in. So a lot of what we do, we try not to compete with industry. I think industry does a very good job at what they do, and our job is mostly to kind of expand the science that we can do with that, and try and do new types of non-incremental science. That's why also we're moving from initially we're doing a lot of single gene type of synthesis, which essentially can be done very, very well by industry, to the more challenging larger pieces of miniature libraries. I think eventually industry will be able to do that also very efficiently, and then we try to move ahead to do something else, just trying to push the field further. I think synthesis will definitely become a commodity in the next few years, but the design of how to build constructs that will work biologically is something that will still need a lot of input from institutes like ourselves and some of our collaborators. So we have another question from Munir Laland. So this question is, how would you use your capabilities to screen for synthetic approaches to improve product tolerance? So I think that's an area that is usually addressed by mutagenesis more than synthesis. But with applied synthesis, for example, if you thought there were sets of pathways that might improve the tolerance, and these pathways come from different organisms to the one where you actually want to develop the tolerance, we could just synthesize and perhaps assemble either individually or combinatorially these pathways and transform them into your cells, and then have some sort of high throughput screen for tolerance. I think in the case of tolerance, the high throughput screen is very simple because you're just looking for survival, so that should work pretty well. But it should all start with a hypothesis of what are the kind of pathways that you think might confer the tolerance phenotype. There's a new question from Andy Schumacher, and his question is: are there challenges associated with translating synthetic enzymes screened in cell-free NIMS type assays into industrial applications within microbial hosts? That is a very good question. And yeah, there's always a difference between in vitro and in vivo performance. Actually, for the specific applications that we were looking for in terms of biomass construction, it's also kind of an in vitro assay, which is basically going to incubate the biomass substrate with enzymes that we provide. Moving them in vivo is a whole different problem, and I think the way to approach that is by having a diversity of enzymes that you synthesize. So if one doesn't work, hopefully one from your set will perform very well. So I think the key here is just tailoring your screen to what you want your final output to be, if that makes sense. So I think that there are no more questions coming in. So I just want to thank you all once again for your attention, and please feel free to contact me if you have any questions, or if you are interested in the CSP product. So thanks very much, and I put my time.
Thank you, everyone, for attending. Thank you, Sam, very much for this excellent presentation. And we will be following up with you in a follow-up email where we'll be distributing the Q&A document so that you have it for your reference.